Okay, so in the first talk, I presented you with some problems, some weaknesses, some threats to the DNA data storage chain, and in this work, what we've developed is a first confidentiality solution for DNA data storage that is based on biomolecular operators, and we've also done a biomolecular proof of concept. So with the same team and helping from the molecular biology front are Dominique Lavenier and Julien Leblanc. So I will skip the introduction. As I've said, during the risk analysis, there are threats at all steps, particularly concerning confidentiality. So our challenge has been to ensure the long-term confidentiality of data stored in DNA molecules. And to do so, I will present our biological security solution. So our idea has been to reproduce digital encryption operations in the biological world to ensure a stronger security that is based on biology. And so we wanted to manipulate DNA, and to do so we have identified 6 classes of biomolecular operators that modify the DNA, the structure of DNA, or modify the content of a DNA pool. And so the first class includes the modifications of DNA ends, for example, enzymes that can remove sticky ends of double-stranded DNA molecules. The second class includes DNA cuts, so using restriction enzymes. Several hundred enzymes recognize different restriction sites, such as the restriction site that's framed in red, and cut them in different ways, with these enzymes all having different yields and parameters. The third class regroups ligation operators, so it's possible to ligate here in this example, complementary sticky ends of double-stranded DNA molecules with enzymes. We've also identified the fourth class, which is modification of DNA bases. Here, with methylation, it's possible to modify bases A to MA bases. So to have an alphabet that's bigger than just bases A, C, T, and G. And the fifth class is the amplification by PCR. So using PCR, we can exponentially amplify the amount of DNA. And the sixth class regroups separations of specific molecules. For example, the molecules presented here are separated according to their extremities, their ends. So in total, we have identified more than 30 operators that can be used to modify the structure of DNA molecules. And based on some of these operators, we have designed 2 more advanced functions to modify the structure of DNA. So the first one is the biological rotation. This biological rotation takes as input a double-stranded DNA molecule and exchanges the positions of 2 data blocks, here D3 and D2. So a data block is a data encoding DNA sequence of hundreds of bases. And to do this rotation, we used several operators, cutting, separating molecules, ligations, and PCR amplifications. More precisely, we used 2 cycles of these operators. And the second function is a biological permutation, which allows us to exchange 2 blocks between 2 molecules. So this function is based on the same operators as the rotation, with an additional operator that allows us to modify the DNA ends, and but this time it only requires 1 cycle. So It's important to note that despite the apparent simplicity of these functions, each of the operators have their own complexity and parameters that are specific to them, and that they do not have 100% yield, so each step brings errors and that are specific to them. And so using these functions, those two functions of biomolecular rotation and permutation, we have developed a security solution that conditions the recovery of information stored in DNA on biological manipulations. So our goal is that to recover data, you have to do some biological manipulations. More precisely, if biological manipulations are not done, the consensus step after sequencing is perturbed. Indeed, as a data coding DNA molecule will exist in different forms in the DNA pool, so it will be impossible to do the majority voting at the post-processing step. So our solution relies on two algorithms. The first one is a digital encryption, DNA-Cipher, in which we encode binary data into different molecules with permuted or rotated sequences. And the second algorithm is the reversed version, but in biomolecular form. So it's a biomolecular decryption algorithm in which we reverse permutations and rotations with biological operators. So let me explain the chain. So to write data into DNA, we take binary data, we encode it into a DNA sequence. The result is a sequence D with several data blocks. These copies of the DNA sequence are encrypted using different rotations and permutations. So, as shown in this example, we obtain copies of the encrypted D sequence with blocks at different positions and of different sizes. So, if we were to sequence the result and to do a consensus, it would be difficult to do it by majority voting. So those are the sequences that we then will synthesize and store in DNA. And to recover data that is stored in DNA, we have these sequences as input, and we will do some DNA-Decipher, the decryption algorithm, which relies on biomolecular rotations and permutations. And after that, we do the sequencing, and we find that the sequences are aligned, and we can do a consensus. So this solution, this security solution, is a first confidentiality algorithm based on biomolecular operators. And we've decided to do a proof of concept of a biological proof of concept. To do so, we've chosen to try to do the biomolecular rotation, which is the more complex of the two functions we developed. So to do so, we took some binary data that we encoded into a digital 400 bases DNA sequence D that we divided in 4 data blocks. And then we applied a digital rotation to it to obtain this sequence with the blocks D2 and D3 that are exchanged, and we synthesized the result. So then, to decrypt this sequence, to inverse the rotation, we developed a biomolecular rotation protocol in 19 steps. So as we see here, the goal is to reverse to rotate the D3 and D2 data blocks. So this protocol diverts for the first time the use of biological operators for security purposes. And it's important to note that even if it appears really simple and is really simple in the digital world, in the biological world, every reaction has its own yield, brings a lot of errors. This protocol also cost us a good amount of money and took several hours of manipulations. So what were the results? After sequencing, we sequenced the results of our biological rotation after 19 steps, and we wanted to obtain as many decrypted molecules as possible. So they are presented in green in the table and represent 14% of molecules. And so we were happy to find that the decrypted molecules were more present than the encrypted ones, and that the decrypted molecules were the second most frequent after small isolated D4 data blocks that are easy to discard. So thus we've proven that it's possible to isolate the decrypted molecule by manipulating DNA for security purposes for the first time. So we did a proof of concept of biomolecular rotation. And so this is first result with the use of biological tools diverted from their original framework, which can be optimized, and we've already thought of some optimizations to get a bigger percentage of occurrences. And so, to conclude, we've developed two biological functions of biomolecular rotation and biomolecular permutation from several elementary biomolecular operators. We've also developed a security solution which requires biological manipulations to decrypt DNA. And finally, our final result is a first of its kind proof of concept of a secured DNA data chain with a biomolecular rotation that modifies the structure of a data encoding DNA molecule. So thank you very much.
Are there any questions?
Thank you very much for your talk. It was very interesting. I was just wondering if instead of using a molecular operator you described, you could use a complete bioinformatic approach, for example, using like a specific sequence pattern.
Thank you. So the question is, okay.
Like so if you could develop an algorithm that could, instead of using like molecular approaches that are very challenging, only develop a software that could do the decryption.
I think we could. Our goal here was to force an attacker that wants to retrieve data to do these biomolecular, biological manipulations to make it difficult for an attacker, or even impossible for an attacker to retrieve data. So for now, what I've presented is a simple first version. The goal would be to do many more, to develop a security algorithm with many more manipulations, and so to make it impossible to do it in bioinformatics.
Okay, so that you would require, for example, a machine that could do the automatic molecular rotation and something like this?
Yes, yes, that would be the goal, yes.
Okay, thanks.
Thank you.
Thanks. Are there any other type of operators that you imagine to implement?
Of course, for now before imagining some really complicated algorithm, we wanted to test that just a simple rotation could be done. So this was really the first step, but we could imagine many more operators to complexify the algorithm. And so yes, I don't know if you wanted me to mention...
Yeah, if you had one more example, for instance. Or not, if you don't have, it's okay.
No, outside of the 6 classes that I mentioned, or?
Yeah, or combining for this...
I think combining with base modifications such as methylation or some things to make the algorithm more complex, and it can also hinder some cuts with restriction enzymes if some bases are methylated or not. So we have many perspectives to complexify the algorithm.
Thanks.
Thank you.
Thank you. So in the proof of concept you've shown, you've got only one biomolecular rotation, but I think in the previous slide you showed that you would apply many rotations, have many different molecules to synthesize, and then how would you manage to decrypt like a single consensus sequence if you had to recover many different rotations from a single pool?
The goal is to use for each different rotation, different restriction enzymes. So, and also to use specific restriction sites, so that when we cut one molecule to apply a rotation to it, the other molecules are not affected. So we use the fact that there are several hundreds restrictions enzymes that are available to do that.
Okay, thank you.
Any other question? I've got one on my side. Did you consider, because you present the security at the digital level and the security at the biological level completely independent, did you consider that you could use the biological operations for and bring the two? Let's say the biological operation will give you the code word that will allow to decrypt the data. Did you consider this kind of link between the biological operation and the digital world, which would be, as I can imagine it, more secured or provide a strong security because you've got the power of all the digital encryption and the digital security, but is dependent on something that is linked to one of your biomolecular operation. Did you consider this?
Yes, we've considered it. I think here I present security from the using only biological manipulations, but of course we would, in a real life scenario, we would combine both digital and biological solutions to really to get the most security.
Okay, thank you very much.
Thank you. And I just wanted to mention that this work and the work of our team continues with 2 PhD students, Marc Canev and Hélène Gagné, in the cyber health team at IMT Atlantic. So thank you.